team will be Bangalore.
Okay, so hi, I'm going to be representing the NCBS team from Bangalore, now called Bacterial. And so our team basically grew out of a workshop we had in January, starting in January, where we got together a lot of people, people from industry, engineers, lawyers, a couple of artists too. We talked about different things, learned about synthetic biology, and we had these three basic projects. And I shall talk about the first two. So one, we wanted to control chemotaxis in 2 dimensions. The second project involved synchronization between cell cycles. And the third one is a UV switch, which will be there in the poster, but I won't really go into it because of time issues. And so I should just point out that the first project is a very simplistic one, but we addressed several practical difficulties in implementing it. Whereas the second one was a lot more complicated, and we actually got somewhere in that, so a bit counterintuitive. So moving on to the first project, we essentially wanted to achieve control over the 2-dimensional motion of bacteria on the solid surface of an agar plate or something. And we just go into a slight brief overview of the chemotactic process. So at a microscopic scale, chemotaxis can be looked at as a series of runs and tumbles, where runs are directed motion and tumbles randomize the direction of the next run. And as the cell goes up a concentration gradient of an attractant, run lengths increase in that direction, and that's how you get a macroscopic phenomenon of chemotaxis of gradient. And looking at the machinery of this involved in this process, you have a chemosensory receptor sitting in the periplasmic space on the domain. It's a transmembrane receptor. There are many types of these, but we are going to focus on TAR and TSR. TAR recognizes aspartate, and TSR is for serine. And there's a whole series of interactions within the cell, and I'm not going to go into detail here, which finally talk to the flagellar motor and either induce tumbles or cause runs. Now could we hack into the system and get what we want, that is 2D motion out of this? But first we need a physical setup in which we can, an arena where we can have this 2D motion taking place. So suppose you have 3 chemotactic gradients, so aspartate and serine, which are orthogonal with each other. And a glucose gradient in another dimension. So you need 3 positive vectors to achieve this 2-dimensional motion. And if you put all of them together in this plate, and now we put the condition that we can actually change the responsiveness of our bugs to aspartate and serine gradients, whereas the glucose one is a default gradient. So we have 2 points of control in this case. Then we hope to achieve 2D motion. And these are the constructs we are going to utilize, trying to utilize to achieve this inducibility to these 2 gradients. So in the first case, you have the TAR receptor expression being controlled by the lac promoter. CFP provides a transcriptional readout of the levels of transcription of mRNA. And the host strain is U1250, which doesn't have TAR or TSR, which is a knockout strain. And the other construct is TSR from this thing, receptor with an RFP readout under control of TET promoter. And there are 2 basic motion control strategies that we could use to get from the colony from point A to point B. One is the polar strategy, so the polar coordinates, where you can induce responsiveness to these 2 gradients at the same time. And this is basically a displacement vector between the 2 positions. The other case is where you use a Cartesian strategy. So you go along the axes. So basically sequential induction to these 2 gradients. So first you make it responsive to aspartate, so it moves down, and then you make it responsive to serine, so it moves to the side. And how we choose between these 2 strategies is is based on the nature of the chemotactic response when you change the receptor concentrations. So in a single gradient, suppose you could increase the receptor concentration by putting different amounts of IPTG, and you have a starting point here, and you just let them go after and watch them up and after a certain point of time. So if there's a linear response, you would expect that chemotactic ability increases with increasing receptors on the surface. Whereas if it's an all-in response, there's a certain threshold beyond which you either get a response or you don't get one. And then a dual gradient setup, because that's the real case here. If there's an all-in-one case, dynamics situation, you would expect that if you have bacteria starting off at the center of a dual gradient setup, you would expect them to fragment and a few stochastically moving to one side and the rest to the other side. And say this is a stronger gradient, then we can have more bacteria here than there. If it is a linear dynamics case where you're using there's no receptor interaction taking place, then you have the whole group as a whole, the group as a whole moving towards one side. So again, all cases requiring linear dynamics would require a polar strategy. And Cartesian dynamics could be utilized for anything, but it's more difficult to implement this. Just some induction results where we just put IPTG, ATC ejected fluorescence. So this is a graph with RFP fluorescence on the y-axis and RCFP on the x-axis. Each dot is the fluorescence from a single cell, so microscopic images. And you can see that autofluorescence at a certain level. Basal expression without any inducers is clustered over here. When you're at IPTG, you see CFP fluorescence increasing and not RFP, so that's fine. But when at ATC, we found that, surprisingly, that CFP fluorescence increases in relation to RFP fluorescence. That is a little strange, so we have to sort this problem out. There's cross-induction happening. And what does this mean in terms of actual chemotactic ability of these cells? Is that, so we need some assay for chemotaxis to test this out. So if you have two gradients independently, here in this case, you start with bugs in the center, these two gradients. You'd expect that if you have no inducer, nothing really happens. When you add ATC, you should expect TSR expression increase and hence it becomes responsive to the serine gradient. And the opposite in the IPTG case. And when you add both inducers, you should find that bacteria are responsive to both gradients. And this is a test of our hypothesis. So first, just some ways we wanted to set up gradients. So in this case, you have a bridge setup, which is a microscopic assay for chemotaxis, where you have a thin layer of soft agar on the slide, and you put two blocks of hard agar, one of which contains a chemoattractant, so that's the source, and the other one is the sink. And between these two is where the gradient is set up in this bridge layer. And this way also inoculate bacteria, and we'll look at them under the microscope. And we just wanted to check if it, from the gradient, so you put rhodamine in one block and nothing in the other, and we found that you do get a gradient being set up. But maintenance over long periods of time is difficult because we had drying up of this setup, of moisture from this case. Another thing is the slant plate. So this is something which you could use on a plate. So if you put a lower slanted layer of hard agar containing chemoattractant and overlay it with a soft layer containing nothing, then by diffusion you get a gradient set up over some point of time. And this is something, it's not come out very clearly here, but there's a potassium permanent gradient set up in this case. And this is something which kind of worked pretty well, so which is an agar plug assay, which we adapted from, which I think Jim Brown at Cambridge had told us about. So if you put two spaces on the slide here, and you put a block of agar containing a chemoattractant in the middle, and then you overlay it with a cover slip, and then you fill the rest of the space up with E. coli suspension. You'd expect that after, and if you image this edge, you'd expect that after some point of time you'd find bacteria accumulating here. And this is an image is taken with over a time period of 30 minutes or so. They aren't really accumulated in this scenario, so this is the agar edge. And this image is taken over 1.5 seconds, so you can see runs as streaks across the image. You find that the runs increase in the general vicinity of this, become longer and increase in frequency over the vicinity of this agar layer. So we still have to tweak this assay to get it exactly right. So we still have to establish that our constructs can rescue chemotaxis, and we have to find out how this happens, the exact dynamics in which this occurs. Then we have to resolve this cross-induction issue, fine-tune the plug assay, do the dual gradient experiments, and we want to construct a model which incorporates different receptor interactions and then see, whether we have an all-in-all dynamics situation or linear dynamics situation, and then decide a control strategy. So let me move on to the second project. So essentially, if you have many oscillators oscillating out of phase, and you have some weak coupling between them, over time you get synchronization between these oscillators. And how? So we wanted to test out whether this works in the case of cell cycles. So in that case, the co-oscillator is the cell cycle itself, because you have periodic event, you grows, divides. And how we wanted to couple it. So this is a co-oscillator. We wanted to couple it using the quantum sensing machinery from Vibrio. So Lux I, autoinducer Lux R. And how would we feed this back in a cell cycle? That's the actual completion of the coupling cycle. Is by using DNA, which is known to modulate cell cycle. So basically oscillation will be set up here, transmitted through the processing machinery outside the cell to other cells, and then fed back into this using DNA A. I'll just get into this in more detail now. So we have module assembly, so these are the parts. I'll just get into them in detail. So in the first case, this is the oscillatory sender module. So what it is just a aryl cyclase promoter, which is a PCY promoter. And this has been found to oscillate with the cell cycle, with the phase cell cycle. So what we have downstream of that is a Lux-size CFP fusion protein. So CFP just so that you can watch it oscillating. And this catalyzes synthesis of autoimmunosal, which is then diffusible across the cell membrane into the external medium. Then an AI degradation module just increases the peak to trough ratio of these oscillations because you decrease the accumulation within the cell itself. And this is under the control of PTAT promoter. These are various controls we've put into the case, so you can see what happens with and without AIA, etc. This is an enzyme that degrades autoimmunosal. There's a receiver module. We have Lux R, which binds to autoimmunosal, and then I'll explain what happens after that later. RFP provides the signal, which is non-oscillatory, because it's under the control of an aerogenous promoter. So you want to compare oscillatory signal from the PCY promoter to a non-oscillatory signal from a regular promoter. Now the oscillation readout module, where what happens is the Lux R binds to autoinducer and then triggers promotion from the P-Lux promoter. So you have YFP gene downstream of that to see whether we're receiving oscillations which are going through the chemo sensing machinery. And I'll explain what IPTG does in the next step, this lac promoter upstream of that. So then, this is how we feed back into the cell cycle. We can actually try to slow it down. And what we have here is the same construct which is here in this case, except that instead of YFP, we have a PX site. And the PX site sequesters DNA A. And DNA A is required for cell cycle progression. So if we sequester it, we tend to slow down the cell cycle. And this is, our hypothesis is that if you have transcription going on from P-Lux promoter, you knock off DNA A from these sites as you're transcribing through this coding, the coding region is PX. So it's kind of crazy idea. And an IPTG is just used to, because we don't know the dynamic range of setup. So we just increase the general transcription level to different amounts so that we are within the dynamic range of P-Lux transcription. And so as you can see, because it's a modular system, we can actually design readouts at each stage and controls to see that everything is working. So just a brief note about characterizing the parts. So this is the oscillatory promoter, and as you can see, so what we've plotted on the y-axis, the x-axis, is the cell length, which is correlated to the cell cycle. And it should go up to 1, but we've just taken this whole data set and reproduced it to the right of this whole thing, so that you just want to see that oscillations come back to the same level it started off with. And what you can see here is that as a function of cell cycle, you do see oscillations from the PCY promoter. And RFP is just this transcription levels from basic promoter, from inducible promoter, which is constant throughout. But what about transmitting signals to the processing machine and receiving them? Now, what we find is that the oscillations are preserved even after this step. In fact, the peak to trough ratio seems to have increased. And that's kind of surprising. We'd expected it to be dampened. So what seems to be happening is that it could be that there's some cooperativity in the binding of Lux R to the P-Lux promoter, but we're not really sure about that yet. Now, the final piece in this whole network is DNA A and the PX sites that I mentioned before. And what the basic test to see if the PX site actually going to work is to put this construct into a cell and just see if it slows down cell cycle, so growth rate. So if you do a growth curve with the bacterial with this construct and with nothing else in it, without a construct, and then if you induce transcription type through IPTG, you would expect to see different results in terms of growth rate. But we found that there is no result. This doesn't really slow down the cell cycle at all. And when we looked at this bacteria under the microscope, we saw that DNA, I mean, there was a problem with DNA sequestration. So it was getting, division of DNA, daughter chromosomes was affected. Segregation was affected, but somehow the rate of growth is not affected. So this is a bit intriguing. So finally the conclusion is that from this particular project is that we have a part which gives us oscillatory POPs output. The upside CFP fusion is functional. Oscillations are transmitted without degradation through the column sensing machinery. But pH alone is not sufficient to affect cell cycle progression. So we are looking at other ways to feed back into cell cycle and slow it down. Just a short note about, so what happened in the earlier case is we had a lot of parts and it took a long time to put them together. So actually cloning all these various parts, especially for cell cycle machinery, cell cycle synchronization problem, took a long time. So we were looking at faster ways of doing this. So is there a single step method of putting all these parts together? And which is scalable, maybe 100 parts, 200 parts, whatever. So this is a strategy we came up with, where you have each part on a plasmid, and this we can linearize, I mean, we make it single stranded through the M13 system. And then you have these staplers, which are basically oligonucleotide linkers. And each part, each plasmid for each part has a unique site, which is recognized by one end of the linker. And then it joins it to the next site, next part, etc. You can just keep on going on like this. So basically what determines the sequence is this sequence combination you put on these linkers. Then we use type 2 restriction enzymes, so that and we have recognition site at another site over here, so that you can make a cut exactly at this place. And this will give you seamless thing. So you can actually make fusion proteins, etc. this way. You don't have any leftover restriction sites at the end of it. And yeah, so we were just trying something out with just to see if with the type 2 restriction enzyme whether this thing works or not. And you can see the results on the poster we have out there. And yeah. So we just want to acknowledge Reshma for all the help she's given us. Panika in NCBS for help with the M13 strategy. All the participants of the workshop. And cloning services provided by Bangalore Gene company in Bangalore. And funding from NCBS. And the next step we plan to focus on DNA and protein sub-assembly. So have a look at the website. That's it.
So we have a few minutes for questions.